Hi everybody again. We're going to have a bit of a look at something called the limitations to cell size. And what this has to do with, as it outlined by the dot point here, has to do with surface area to volume ratios of cells, but also of other parts of plants and animals as well. Now, this is particularly central around the ideas behind diffusion and osmosis, and this particular idea of surface area to volume will come up a number of times again, both in the prelim and the HSC. Now, before we go on, I should say that the limitations to cell size is not a standalone topic, it's more or less rolled into the ideas behind diffusion and osmosis. So if you don't understand those just yet, can I suggest you may just go back and review the previous couple of lessons to really make sure you have a good handle on it. So the first thing that we'll do is we'll have a look at the idea of surface area to volume, just looking at a single cell. Now, one of the things to bear in mind is that cells need to have things cross the membrane. Okay, they can either go directly through the membrane or through some of those pore channels or proteins which allow substances through. But either way, what we need to be aware of is that cells need to get the things that they want into the cell and the wastes that they need to get rid of out of the cell. Now, all of these things have got to pass through the cell membrane. And when we're thinking about a cell, the way we can look at the membrane is that is the surface of the cell, and so obviously that'll be the surface area of the cell. The contents of the cell, so everything inside the membrane, the way we could look at that is being the volume of the cell. So I'm going to have a slight attack of mathematics here in a moment, but what we can see is that the surface area of a cube is determined by the formula 6 times length times height, and the volume is determined by length times breadth times height. And the surface area to volume is just the ratio is just surface. So let's have a look at the surface area to volume ratio. So if I start off with my 1 centimetre cubed here, okay, 1 times 1 times 1 gives me a volume of 1, but the surface area, it's got 6 faces which are both 1 by 1, so 6 times 1 by 1 gives me 6. So the surface area to volume of the small cube will be 6 to 1. Then if I go to a bigger cube, say 3 centimetres, all right, my surface area will be 3 by 3, 6 of those, so 3 by 3 is 9, 6 of those is 54. And then if I do 3 times 3 times 3, the volume is 27. So 54 divided by 27 gives me a surface area to volume ratio of 2 to 1. But then if I go to 6, and then I go 6 by 6 by 6 for the volume, but then at the same time, my surface area is going to be 6 times 6 by 6 as well, so that'll all equal 1. So what we can see quite plainly is that as a cell gets bigger, the surface area to volume ratio progressively decreases. Now, what are the implications of this for a cell? Well, basically what it means is that for the amount of volume it has less surface area, so it has less ability for each unit of volume to get the things that it wants in and the things that it needs to get rid of out. And so let's have a bit of a look at this again, but this time we'll have a look at it in terms of something a bit more like a cell in a sphere. So on our left hand side here we have a small cell which begins to grow until it gets to a radius of 3 times its original size. And so what happens here is that if you compare the surface area to volume ratio, so we can see down here the small cell has a surface area ratio of 3, but the bigger cell has a surface area to volume ratio only of 1. And so to counteract that, the thing that the cells do is as they get bigger, they begin to divide. And when they do this, they can restore that surface area to volume ratio. So let's have a look at that example again, but just consider it from a slightly different angle. With our small cell here, what we can see is that it has 3 units of surface area to supply it, and we can liken this to a funnel. And because it has a much greater surface area to volume ratio, the funnel is much bigger, so more stuff can go through and into the cell. However, what this means for a bigger cell is that because of each unit of volume, it only has 1 unit of surface area, so the size of the funnel per unit of surface area becomes much smaller. And so hopefully this shows you a nice way in how it becomes difficult for the cell to service its internal needs because it just doesn't have the ability to get the stuff in as quickly. So as I was saying, the good part about having a large surface area to volume ratio is that you can get things into the cell quickly and you can get things out of the cell quickly as needed. However, there may be some things that you want to hang on to but you're just going to lose anyway. And these may include things like water, it may include heat, or it may include other things, other nutrients or something along those lines if they have a surface area which is too large. And this is one of the things about biology, is that you follow some basic rules, but then every once in a while you have to compromise, you have to balance them all out to make sure. Now on the converse side to this is that if you are too big, and as I was saying before, you may have a hard time losing that heat or losing wastes or something like that. And this is shown quite beautifully by our slightly overheated dog here laying in the ice. So this once again emphasises the idea that you actually have to come up with a balance. So you have enough surface area that you can keep yourself alive or keep the cells alive. Now, surface area to volume ratios are not just important for cells, but they also become important for individual animals as well. And when I was trying to think about some of the relevant examples that I could give you, this one isn't an Australian one, but it does highlight it very well. So our first example here is we have the Asian sun bear. So this one is actually a tropical bear. Now have a look at its body shape and its size compared to something which lives up in the Arctic, so the polar bear, okay, one that lives in Siberia. This is the Kodiak bear, and then you also have the brown grizzly which lives in the northern parts of America and Canada. And we can actually see here that this one, because it has living in a tropical climate, we can see that it is a smaller in size, and so as a result it'll have a higher surface area to volume ratio, and that's quite good for losing heat. On the other hand, however, these bears in colder climates are much bigger, and because of that their surface area to volume ratios are smaller, and so it actually retains the heat. But in this particular case, the size and the reduced surface area to volume ratio is at quite an advantage for them. Now another example of where shapes can be modified as well to either minimise or maximise surface area to volume is in leaf design. So down on the bottom here we have a fern, and you can see here that the leaves are quite flat, and that is quite good because what it means is it has high surface area to catch light, but it has low volume as well, so the light can be made available to all parts of the leaves which are capable of photosynthesis. However, on the downside, it means that there could be water losses which could be quite high, and so it's for this reason that ferns are generally found in moister climates. On the other hand, we have this particular plant up the top. Now this is a casuarina, so you may have seen these when you're walking to the beach. And the casuarina here, you can see that it doesn't have normal leaves as such, they're more like needles. And the reason for that is because they live in sandy soil, and so the amount of water that they can trap after a shower is not a lot, because sandy soil, the water tends to go through it very, very quickly. So these are adapted for a drier climate, and as a result of that, what they've done is that they have reduced their surface area to volume ratio by making their leaves cylindrical, and as a result they can save water. The trade-off with that, however, is that they are not as efficient as in photosynthesis using light as the plant on the bottom would be. So we're now nearing pretty much the end of this particular presentation, so I just thought I'd reiterate some of the key points that would be worth knowing for you. So make sure that you can define what surface area to volume is, understand why cells need a large surface area to volume ratio, and then if you want to take it one step further, explain how for different organisms surface area to volume will change depending on their environments and their needs. Now I'll also have a reading up on Moodle as well, have a bit of a look at that. And then finally, I will actually give you a separate exercise which I'd just like you to complete on your own. So I guess in terms of note taking, if you can make sure that you have all of these things done, you're well on the way. If you would prefer something a little bit more structured, then maybe use these particular, the information on these next 3 slides, and these will also make some quite handy notes for you that you can use to revise. All right, so just talking about surface area to volume here, and where surface area applies, and that's the membrane, as opposed to volume where it is the cell contents. So have a go at that particular set of questions. Now this particular question is more about applying what you have hopefully learned in the last few minutes about this. So we've got a student here which is making plasticine models. The cells should have different surface area to volume ratios. So when you're thinking about this, talk about not only whether the cell should be small or large, but also consider the shape. I'll just give you an example on the next slide here. So what you can see here is that the depending on the size, this one's shown quite nicely for you on the bottom here with the cubes. So you can see as the cells get bigger, the surface to volume area decreases. But the other thing that you may want to consider is the shape of the cells too. So should the cells be sort of cubic or spherical, or are they better off being flat? So have a bit of a look at that. Finally, this is the last bit of application that you can do, is have a bit of a think about these particular examples down the bottom here. So 1, 2, 3, 4, and try and link them up to the shapes across the top here. All right, so an animal living in the Arctic tundra, we talked about that with the bear. We also talked about a tropical rainforest plant. So for example, there probably wouldn't be a problem with a water shortage for that one. Then you might want to talk about a tree leaf, all right, where small amounts of rain fall infrequently. So you might want to think about your answer in B and see if you can extend on that and take it to C. And then finally, an animal where there are warm summers but cool to cold winters. So the first 2 shapes should be fairly obvious to you. If you want to suggest an alternative shape, you can do that. The emphasis will be on your explanations for it. All right, so there's a fair bit of work that can be done on this, and like I said, it's very, very important that you get a handle on it, because this is stuff which will come back numerous times in the prelim and certainly will again in the HSC. So good luck.